Janos. So I have the pleasure today to talk to you about our case study on the persistence of Listeria monocytogenes in the food processing environment. So again, I'm going to briefly introduce the FoodSafeR project. So it is a 4-year project, currently in its 3rd year. And we are establishing the FoodSafeR digital hub, as Christina nicely presented to you a minute ago. And this is the QR code. If it's working right now, we operate under the hashtag food safety is everybody's business. And if the hashtag is not working, please come see us tomorrow at the EU Project Marketplace. We will be there to present more information about the FoodSafeR project and hopefully invite some of you to join the platform. So the hot and very difficult topic of persistence. So persistence of Listeria monocytogenes is defined as the repeated or long-term survival of Listeria monocytogenes in the same food processing facility over long periods of time. And so this is introduced through different raw materials, different food products, and they can become established in different niches or harborage sites within the food processing environment, which could lead to the contamination or recontamination of the final food product. And as we've heard today repeatedly, Listeria monocytogenes, despite its low prevalence, can have quite an effect on the health of human beings. But the challenges come when we try to study this organism, in that we have no concrete or defined definition for persistence. We use various typing methods which complicate comparison across studies, and phenotypic and genotypic studies have not yet been able to link specific genes or traits to this phenomenon of persistence, leaving a lot of questions in our studies. So in FoodSafeR, we try to take a combined approach to looking at this difficult topic. So we're drawing upon historical information from the literature, also from existing databases and resources we have available to us. We've also conducted a cross-country sampling campaign with our partners in the project, and together we're trying to form the Persisters database as a resource for the community to predict, based on the knowledge we do have, which strains might be persistent and which factors might play a role in this. So I'm going to try to unravel some of these topics today with you. So we're going to start with the first part, which we take a look into the literature. And we wanted to know, first of all, is persistence a widespread or type-specific phenomenon? So Listeria monocytogenes, many of you already know, are defined into sequence types, which are then clustered into clonal complexes. And across the literature, we see 2 different lineages, lineage 1 and 2, and these are assigned into different clonal complexes. And we see some slight patterns here, where we see clonal complex 5, 1, 6, 9, and 121 as the most commonly isolated across the lineages. And we see that clonal complex 9 and 121 are more commonly shown to be persistent than non-persistent. But what we can really take away from this is that persistence is a widespread phenomenon. Most sequence types are capable of this in the food processing environment. We then wanted to look into the literature to see, are there any traits that can be robustly linked to the phenomenon of persistence? And what we commonly see across studies is that disinfectant resistance, stress tolerance genes, and mobile genetic elements are more commonly associated with the persistent state than in the non-persistent state. But what we do see, this is all clonal complexes combined. When we take a deeper look at a specific sequence type, we see a slightly different pattern in the genes that are associated with persistence. So this requires perhaps a sequence type specific approach to looking at these biomarkers. So now I'm going to take another step into a database we have available thanks to the National Reference Laboratory in Austria. So we have the Austrian Agency for Health and Food Safety, the AGES, And they're responsible for collecting all of the Listeria monocytogenes isolates and genome sequences for the country. And they have amassed a database that's continuously growing with over 17,000 Listeria monocytogenes genomes. And this is an incredible resource and opportunity for us to look for patterns in our genome data. So what we've done here is we've collected a subset of the companies that regularly report to IAS. So we have a list of 20 companies here with submissions of genome or of isolates between 2018 and 2023. And this database was narrowed down then to 1,838 genomes from the food processing environment, representing 28 different sequence types and 93 different core genome MLST types. So we then applied a working definition for persistence based on the genome, because we had very limited metadata to work with. So we decided our definition here would be a repeated isolation, so at least 3 times of the identical isolate from the same facility over at least 2 calendar years. And the identical isolates should have less than or equal to 10 SNP differences based on the whole genome SNP analysis. And this is a slightly more conservative approach than what's widely used in the literature. So on our first glance, looking at this database with this definition, we were able to classify 757 non-persistent genomes and 1,054 persistent strains. And if we take another look here, we have the 2 different lineages and the different ST types. And what we can see is some of the canonical ST types like ST8, ST9, and a few, so we're missing ST121 here, interestingly enough. But what we can see is, again, across the different sequence types, we have both persistent and non-persistent strains. Then I'm going to just briefly summarize this because it's hard to read. We started looking into potential biomarkers within these genomes, and what we can take away from this preliminary analysis that's still ongoing is that we have some toxin-antitoxin genes, ABC transporters, transcriptional repressors, CRISPR-Cas genes, and some of those more canonical cadmium-zinc transporters and arsenic resistance genes that are more related to the persistence phenomenon than non-persistence. But we also have hundreds and hundreds of hypothetical proteins and things that might be associated but have not yet been characterized. So the takeaways here are persistence is a widespread phenomenon across many different sequence types, but we do see patterns of mobile genetic elements, some toxin-antitoxin systems, but these require further investigations, and we should take a more sequence type specific investigation here. And the one major factor that we're trying to incorporate now, but has not yet been fully realized, is the role of the environment and other factors in this persistence phenomenon. So now I will go into our sampling campaign. So across our 3 major sampling partners, so we have Greece, Spain, and Austria as our 3 major sampling partners. We went out to food processing facilities. We had a total of 8 different facilities, and we sampled 50 samples at each sampling time point at 3 different time points, and tried to sample before and after cleaning and disinfection. And at those sampling time points, we not only looked for Listeria monocytogenes, but also Listeria species. We have now finished all of the whole genome sequencing of these isolates, and we also started to look into co-occurring microbiomes from these sampling locations and tried as well as we could to get the highest quality metadata possible. So these are some of our initial summary data from our sampling. So in total, we had 3 dairy facilities, 1 fish facility, and 4 meat processing facilities. We took a total of 1,186 194 swabs. And these were distributed as such across the 3 different facility types. We had roughly half and half from food contact and food non-contact surfaces. And we had a bias kind of towards the pre-cleaning and disinfection, versus the post-cleaning and disinfection phase. When we looked into the percentage of positive samples, we had 26% of our total swab samples positive for Listeria monocytogenes, 23% for Listeria species, and 15% positive for both in the same sample. So they like to co-occur with one another. And when we take a closer look at our pre- and post-disinfection, our ability to remove the Listeria monocytogenes, we see that across Listeria monocytogenes, Listeria species, and the co-occurrence of both, our cleaning and disinfection procedures may not be completely effective because we see roughly the same percentage of the total swab samples positive pre- and post-cleaning disinfection. So our step now is looking deeper into the genome data that we have available to us from our whole genome sequencing. So I think we have a total of about 300 Listeria monocytogenes genomes. And right now, these are the preliminary results from the fish and the dairy facility from Austria. And we can have some evidence here that from our fish facility, we have 27 genomes with less than 2 SNP differences, indicating that they're likely persisters according to our definition. And we have 1 SC9 that's in a different sequence type, but also has higher SNP difference from the rest of the genomes, indicating that it is likely a singleton or a sporadic genome. Then in our dairy facility, we have a set of 33 genomes with less than 7 SNP differences, and this goes back to 2018. So our goal now is to look into the genomes at a deeper level, look into these genomes for genes that could be related to persistence, and also look at the co-occurring microbiome. And finally, our concerted effort here is to build a persisters database that will be posted on the Digital Hub. And this is our goal here is to link our phenotype data, environmental data, and our genome data into a probabilistic framework and see if we can explain or demonstrate how much explanatory power each of these factors has in the phenomenon of persistence. So we have genome data, SNP data, and we're measuring now biofilm information and biosite tolerance of these different strains. We also have our metadata and our co-occurring microbiome data from the sampling locations. And so, just to summarize again, the genes and the phenotype alone have not been able to describe this phenomenon, but hopefully by incorporating some of these other factors and trying to explain the variation in this phenotype, we will be able to get a deeper understanding. And again, looking into some of these hypothetical genes and also a more sequence type specific approach may be necessary, although more complicated in the future of food safety. So I want to thank all the project partners who have been involved in sampling and analysis and everything, and thank you for your attention.